Morning, children. So far, we have discussed the oxidation of fatty acids. Various types of fatty acid oxidative pathways has been discussed, and it was started with the major pathway, the principal pathway, the beta oxidation, which involves 4 steps characterized by 4 different enzymes, of which 2 are oxidative reactions. One is a hydration reaction, the last one is a cleavage reaction, and the product is acetyl-CoA molecules in case of an even chain fatty acid. And odd chain fatty acid, the pathway is almost the same, but the last product of the last cycle will be propionyl-CoA rather than an acetyl-CoA molecule, right? So we have discussed the beta oxidation of both even and odd chain fatty acids. Odd chain fatty acids is rich in the dietary source milk, cow's milk. Okay, then we have entered into a minor pathway which is known as peroxisomal oxidation, which is happening in the peroxisomes rather than the mitochondrial matrix, because peroxisomal oxidation involves the oxidation or the shortening of very long chain fatty acids to the form of short chain or medium chain fatty acid. That is, it ends up in the formation of an 8-carbon octanoyl fatty acyl CoA, which is then entering into the cytoplasm, and further it enters into the mitochondrial matrix for the completion of oxidation, right? So peroxisomal oxidation is again a variant of beta oxidation. It is a modification of only the mitochondrial beta oxidation, right? The difference lies only in the first enzyme. Again, that is a dehydrogenation reaction, that is the oxidation reaction happening, but the product obtained, that is FADH2, which is formed as a result of the oxidation of the fatty acyl CoA, is not entering into the electron transport chain for ATP production. Rather, what happens? The FADH2 releases the protons to oxygen, so that hydrogen peroxide will be formed, and this hydrogen peroxide is then broken up by peroxisomal catalase enzyme, and further give rise to water and oxygen atom, right? So that is how the beta oxidation is happening in the peroxisomes, and they shortens the very long chain fatty acid into octanoyl CoA, right? So we have discussed both the beta oxidation and peroxisomal oxidation. So peroxisomal oxidation is a minor pathway. Now other minor pathways includes are an alpha oxidation and omega oxidation, right? So today we are going to discuss about alpha oxidation and omega oxidation. So what is alpha oxidation? What is omega oxidation? So these are the minor pathways of fatty acid oxidation, which involves alpha oxidation, omega oxidation, peroxisomal fatty acid oxidation, of which peroxisomal oxidation we have already discussed. Today we are going to discuss on alpha oxidation and omega oxidation. So beta oxidation is that oxidative pathway or breakdown pathway or catabolic pathway of the fatty acid, so that the chain is broken up between the alpha and the beta carbon atoms, right? So that is why it is known as beta chain or beta oxidation. So what could be alpha oxidation? What is the difference? It is the alpha carbon atom which is subjected for catabolism, or it is subjected for the enzyme activity. That is why it is known as an alpha oxidation. Of course, it is only a minor pathway, right? The major pathway is, of course, the beta oxidation. In some rare cases, that is based again on the nature of the fatty acid which is subjected for oxidation, the beta oxidation will be replaced. In some cases, in certain cases, the beta oxidation will be replaced with alpha oxidation and omega oxidation. So here the oxidation is happening on the C2 or the alpha carbon atom rather than in between the alpha and the beta carbon atom. Similarly, in omega oxidation, what do you mean by omega? Omega is equivalent to the Z of our English alphabet, right? So alpha means A, beta means B, right? So in case of the Greek alphabets, omega is the last carbon atom. So where can be the omega oxidation happening in the case of omega chain fatty acids? It is on the last carbon atom, which is the farthest away from the carbonyl group or carboxyl group in the opposite extremely carbon atom will be subjected for oxidation in case of omega oxidation. That's why it's known as omega. Omega means the final, the last one, right? So that is the two type of other minor oxidative pathways today that we are going to discuss today. So alpha oxidation and omega oxidation. So shall we start? We shall start the pathways with alpha oxidation. So what is alpha oxidation? What is the peculiarity of alpha oxidation? Where is it happening? It is happening in the microsomes of brain and leave out. If something happens, where some defect happens in the alpha oxidation, it can result in the damage of the brain cells. Clear? The neurons main right? So it is known as it is having, or it is affecting what? It is affecting the brain and mainly microsomes, which is a site. It is a microsomes where the oxidation is taking place, not in the mitochondrial matrix or in the peroxisomes. It is happening in the microsomes. Okay. So it involves, so which are the fatty acids which are subject for fatty alpha oxidation of fatty acids? Those fatty acids where the beta oxidative enzymes cannot have access. Beta oxidative enzymes, that is FAD or fatty acid, FAD dependent dehydrogenase enzyme acts in between the alpha and the beta carbon atom so that the beta oxidation will be initiated, right? So in those fatty acids where the beta oxidative enzymes cannot act on the bond between the alpha and the beta carbon atom due to some reasons. If the enzymes are not able to approach the bond lying between the alpha and the beta carbon atom, those fatty acids are subjected for alpha oxidation. Clear? So some sort of modification happened. So what could be the reason the enzymes cannot approach, cannot give access to the beta carbon atom? Because some side chains are attached onto the alpha carbon atom. Fatty acids. So this is our alpha carbon atom. This is the beta carbon atom, right? It's a fatty. It's palmitoyl CoA. So this is the this is the alpha carbon atom. Easy, right? Suppose propionyl CoA. Suppose I'm just indicating the alpha and the beta. Propionyl CoA. Right? So this is the alpha carbon atom. This is the beta carbon atom, right? This is a functional group, CO, SCOA. SCOA marking COO minus. So it will be left free, and the alphabets are named from this particular carbon atom. So this is the alpha carbon atom, this is the beta carbon atom. So if the enzymes are not able to approach this bond due to some restriction, enzymes not approach fatty acids channeled into alpha oxidation rather than beta oxidation. So what could be the restriction? There may be some side chains attached. So very bulky groups attached to fatty acids are subjecting them to alpha oxidation. Clear? Because the enzymes can act only on the beta bond free because our ultimate aim is to release acetyl CoA. Our acetyl CoA break CH3CO release break the enzymes has to come and act on here. So if the enzymes cannot come and act on here, what will be the ultimate effect? We cannot get acetyl CoA molecules. Some sort of modification happens. So that again the alpha and beta carbon bond is exposed. Expose the modifications. Pathways we call it as alpha oxidative pathways. I'll come to it. Okay. So I've just shown to indicate which is alpha and the beta carbon atoms. So it takes place in the microsomes of the brain. And what is happening? It is that pathway in which the oxidation of the fatty acid with methyl group attached onto the beta carbon atom. If the beta carbon atom is having methyl side chains, methyl group side chains, methyl groups are attached. That is, see, this is a C16 fatty acid. Fatty acid is alcohol, phytol, and corresponding fatty acid is phytanic acid. Right? So phytanic acid is an example. It is a fatty acid, and we can obtain phytanic acid is rich in vegetarian sources, vegetables. Vegetables source are phytanic acid. So if this phytanic acid, see, this is a first functional group. So this is the first carbon atom, this is the second, and this is the third, right? Or this is the alpha carbon atom, and this is the beta carbon atom. So the enzyme should come and act on this particular bond. But the enzymes cannot come because here this beta carbon atom is attached to a bulky group, a methyl group. Side chain enzymes act grip. So what should be done? First, I know alternative. What is carbon dioxide? Right? This remove it. CH2 will be oxidized to carbon dioxide. CO minus. Clear? E bond clear. Appa endu cheyum cheyna idine ange remove edittu the will be removed in the form of carbon dioxide, and this CH2 will be converted into COO minus. Appa idu COO minus, idu alpha carbon atom, idu beta. Appa ivide ni endha CH2 aanu undava. So see ivide CH2 ഉണ്ടാവും, right? Appa ee CH2 now can അല്ലെങ്കിൽ enzymes can act in between the alpha and the beta carbon atom. So such fatty acids which are undergoing such sort of modifications, we call it as alpha oxidation, because it is on the first it is on the bond which is getting attacked by the enzyme is this bond, that is bond between the first and the second carbon atom, or in between the alpha carbon atom is being acted upon by the enzyme. So that is why it is known as alpha oxidation. Clear? So alpha oxidation, it is the oxidative, it's a minor oxidative pathway which is happening in those fatty acids having a methyl group present on the beta carbon atom as a side chain. So that is, if the beta carbon atom is having a side chain like methyl group, then it will be attacked by alpha oxidative enzymes rather than beta oxidation. So beta oxidation, of course, in the beta oxidation can be continued, but the first step is different from that of beta oxidation. So such pathways are known as alpha oxidative pathway. Right? Again, phytanic acid. Phytanic acid is a substance which is present again in cow's milk. ഒരു മിൽക്കിലാണ് ഇത് മെയിൻ ആയിട്ട് again കാണുന്നത്. ഫൈറ്റനൈറ്റ്സ് മിൽക്ക്, അതുപോലെ ക്ലോറോഫിൽ, ആനിമൽ ഫാറ്റിലൊക്കെ കാണുന്നതാണ് ഈ ഫൈറ്റോൾസ്. ഫൈറ്റോൾ വിൽ ബി കൺവേർട്ടഡ് ഫസ്റ്റ് ഇൻടു ഫൈറ്റാനിക് ആസിഡ്. ഫൈറ്റോൾ എപ്പോഴും ഉള്ളത് വെജിറ്റബിൾസിലാണ്, അല്ലെങ്കിൽ ഗ്രീൻ ലീഫി വെജിറ്റേറിയൻ സോഴ്സസിലാണ്, പ്ലാന്റ്സിലാണ്. ക്ലോറോഫിലുള്ള ഒരു സബ്സ്റ്റൻസ് ആണ് ഫൈറ്റോൾ. Right? Appa phytol will be converted into phytanic acid. So this phytanic acid is it's a fatty acid, but this fatty acid is having side chains on the beta position. So it cannot carry out the normal beta oxidative pathway, right? So instead, some sort of modification happens and the alpha carbon is being attacked. Hence, the pathway is known as alpha oxidation. So how is alpha oxidation happening? It is, that is the methyl group blocks. Actually, the blocking are beta oxidative enzymes. Now, beta oxidation is not taking place. Clear? So, it is happening in the endoplasmic reticulum. What are microsomes in fact? And then microsomes, a rough endoplasmic reticulum and smooth endoplasmic reticulum. Have you heard that? SER and RER. While rough endoplasmic reticulum, cell lysis roughness structures are there. Right? Smooth endoplasmic reticulum in the surface appears smooth. There are certain projections in the case of rough endoplasmic reticulum. Our projections can be called as microsomes, and it is in the microsomes that is the alpha oxidation is taking place. Clear? It is not happening actually in the smooth endoplasmic reticulum. It is on the rough endoplasmic reticulum. So it is occurring in the endoplasmic reticulum and the mitochondria of the brain cells and liver. And again, the purpose of alpha oxidation is not to produce ATP. ATP production is not the purpose. Instead, those fatty acids which are having side chains, methyl side chains like a beta carbon, beta oxidation is not possible. So, how can it be catabolized? It remains undigested. Body can use it. The body cannot utilize it, these fatty acids. പക്ഷേ നമുക്ക് എന്ത് വേണം, നമ്മുടെ ന്യൂട്രിയൻസ്, ക്ലോറോഫിൽ, വെജിറ്റേറിയൻസ് ഒക്കെ ആണെങ്കിൽ അതിനകത്തുള്ള ഫാറ്റി ആസിഡ്സ് we have to obtain. So instead of beta oxidation, the body processes the such fatty acids which are having side chains with the help of another pathway which are known as alpha oxidation. So how is alpha oxidation happening? The phytols. Phytols are the actual form of the fatty acids. The phytols will be first converted into phytanic acid. It is the activation. Phytol is first activated to phytanic acid. By this phytanic acid then is converted into phytanoyl CoA. How? By phytanic acid synthetase enzyme or phytanoyl CoA synthetase enzyme. So phytanic acid will be converted first into phytanoyl CoA. Phytanic acid is first converted into phytanoyl CoA or phytanoyl CoA synthetase by the enzyme synthetase enzyme with the consumption of one molecule of ATP with the release of AMP and 2 molecules of inorganic phosphate. So this is phytanic acid. Right? So phytanic acid is a C20 fatty acid. 1, 2, 3, 4, 5. 1, 2, 3, 4. 4 3s are 12. Right? 13, 14, 15, 16, 17. ഇത് ത്രൈസ് ആണ്. അപ്പൊ മിഥൈൽ ഗ്രൂപ്പ് മിഥൈൽ സൈഡ് ചെയിൻ എത്ര എണ്ണം വരും? 3. അപ്പൊ